Hello there. Welcome to Fundamentals of Biology, Lesson 11, where we're going to be looking at mitosis. So this follows on from the previous sessions where we've been investigating cells. So we started off looking at cell structures, and then we moved on to focus on cell membranes. And then in the last session, we looked at cellular respiration. What we're going to look at in this session is the first of two types of cell division, and that is mitosis. The aim of this session is to talk a little bit about what mitosis is and why it's important in growth and repair of an organism. Then we need to dive a little deeper into it and have a look at the different stages of mitosis to talk about what's going on in each one. And then as we go through, I'll explain why mitosis and how mitosis conserves the number of chromosomes from parent to daughter cell. So there's a few different, I think the hardest thing about this topic is really learning the terminology. So there's quite a few new words, quite a lot of new terminology that will come your way during this session and in the next session on meiosis. I strongly recommend, as we work through this, make a note of some of the key words. Then later on, you can maybe jot down the definitions of them. So there's a couple there. So daughter cells is one that you might not have come across before. So I'll explain what they are as we go through. Chromosomes. Well, chromosomes is something that I have mentioned in previous sessions, and it's a word that you've no doubt heard before. But it's important that you know what a chromosome actually is, what it's made up of, what structure it actually is, because that will help you to really understand mitosis and meiosis better. There's also another word there, somatic. So what are somatic cells? So we'll go through and we'll talk about that as well. So there are going to be quite a few words that you need to look out for. And I'll highlight them as we go through. I'd recommend making a note of them, and then during your recap of your notes or during revision or something like that, just write down the word and jot down a definition of it beside it. Understanding the key terms will definitely help you to understand the topic. Okay, so let's make a start then. So what is cell division? To start with, it's one of the fundamental aspects of life. It's one of the things that distinguishes living things from non-living matter. So when you look at a living organism, it doesn't matter whether it's a bacterium or whether it's a human, we know that those organisms are able to reproduce, or they're able to divide, they're able to produce new versions of themselves. If you look at a non-living item, so you look at a rock or a coffee mug or something like that, well, you can say with almost certainty that those two things are not going to be able to reproduce themselves. This ability to reproduce is one of the key distinguishing factors of life. The ability to reproduce is very much tied in with cell division. We also find that the continuity of life, both for an individual and for the species as a whole, is based on the reproduction of cells. So this is cell division. If you want to reproduce, so as humans, we're sexually reproducing organisms. So to enable us to reproduce sexually, we have to produce gametes or sex cells, eggs in females, sperm in males. So we'll look at that more in the next session. That's a process called meiosis, the production of sex cells or gametes. But also we need to be able to grow and develop. So clearly we're not born as fully developed adults. That would stretch the bounds of physics. You can't have one organism give birth to an equally sized organism. It's just not possible. So we give birth to small young, and then they have to grow and develop. Well, how do they grow and develop? They do it through reproducing their cells or through cell division. So this ability to continue as an individual and then reproduce to produce offspring that can continue the species is very much tied in and reliant on cell division, both mitosis and meiosis. So let's give a definition to each of those different types of cell division then. So mitosis, which is what we're going to be looking at in this session, is the division of somatic cells. So somatic cells are our typical body cells. So they are essentially any cell that isn't a gamete, or any cell that isn't an egg or a sperm. Okay, so eggs and sperm are called gametes. Everything else, whether you're talking about a skin cell or a blood cell or a muscle cell, they are all referred to as somatic cells. So mitosis is the division of our somatic cells. And that's what we're going to be focusing on in this session. In the next session, we're going to be looking at meiosis. And meiosis is the production of gametes, or the production of sex cells, so eggs and sperm. An important distinction to make here is that mitosis is the division of somatic cells, whereas meiosis is the production of gametes. So when students are first learning this topic, they often get a bit confused and they will say that meiosis is the division of gametes. It's not the division of gametes, it's the production of gametes. Okay, so those are our two different types of cell division then. Mitosis for our everyday body cells, our somatic cells, and then meiosis for producing our eggs and sperm. And like I said, in this session, we're just going to be focused on mitosis. And then in the next session, we'll have a look at meiosis. So what is mitosis then, and why is it important? In unicellular organisms, so if we look at single-celled bacteria, for instance, when one organism divides, you get an entirely new organism. And this is through a process called binary fission. So you can see up in the top right-hand corner here, this is a bacterium, and then it goes through this process of division, this process of binary fission. And then once it's complete, you now have two complete organisms. Now, clearly that isn't going to work with us because we are multicellular organisms. So a typical adult human consists of somewhere between sort of 70 and 100 trillion individual cells. So there's a lot more cells there to be able to have to divide. Instead, we use cell division, in this case mitosis, for three main purposes. And they are development from a fertilized cell. So every single one of us began life as a fertilized egg. So our mum produced an egg, or to give it its correct name, an ovum. And then our dad created sperm. And then one sperm fertilized one ovum to produce a fertilized cell that we would refer to as a zygote. That is a fertilized egg. But from that point, so you end up with one cell, which is the sperm, one cell that is the ovum. From that point on, we go through a process of rapid development. So we can obviously then turn into a developing embryo, and then into a fetus. And then from there, we turn into an infant human. So all of that is done through a process of mitosis, through rapid cell division. And then once we're born, clearly we're still very, very small. We need to be small for our mothers to be able to give birth to us. And from that point on, we go through a process of growth. And growth, again, is done through a process of cell division, through mitosis, through our cells dividing to produce more cells, to the point where we become an adult human consisting of about 100 trillion individual cells. But as we go through the trials of life, we'll often need to repair ourselves. So you could fall over as a child and break your leg, or break your arm, or skin your knee, or something like that, cut yourself shaving, whatever it might be. Our bodies don't stay damaged permanently. They tend to repair themselves. Well, that process of repair, again, is largely carried out through cell division, through mitosis, the production of new cells to fix that damage. And I've got a little image of a lizard over here, and some lizards anyway are well known for being able to shed their tails as a defense mechanism. And some lizards are then able to actually regrow their tail. Some are better at it than others. other species of animals. So salamanders, for instance, are even better at it, at regrowing lost limbs. But this process of regrowing lost tissue is again carried out through mitosis. So this is mitosis then. We use it for development from a fertilized cell. And then once we're born, we use it to grow. And then as we go through life, we're able to rely on mitosis to repair damage that occurs through just the normal wear and tear of life. But mitosis is only one part of what we refer to as the cell cycle. So the cell cycle is the entire life of a single individual cell from the time it's formed to the time it actually divides itself. Okay, so we refer to that as the cell cycle. And on the next slide, I've got a little diagram that we'll walk through that shows you the entire cell cycle. What we can see here then is that cell cycle. So let's start off down here so we can see these two cells. These two cells have just divided. Okay, so these were one cell, what we would refer to as the parent cell. They have undergone the process of mitosis. So they have split in half, they've completely divided, and these are now what we would refer to as two daughter cells. The fact that we refer to them as daughter cells, they don't have any sex or gender associated with them. This is just a term that scientists coined a long time ago, and now we refer to parent cells and then daughter cells. The cell cycle of these two cells will begin the moment they are fully divided. So we can discard this one for the moment, and we'll just follow this cell here through its own cycle. So once it's produced, it then enters into its own cell cycle, and the majority of the life of a cell is spent in something called interphase. So you see interphase up here. It starts from the moment the cell is produced, and you can follow this blue line. You can see it goes all the way around here, following the arrow, right up into the point where the cell will go through mitosis itself. So where it will divide into two of its own daughter cells. So let's start back over here then. This cell is just entering the cycle, and we can see that the first part of interphase, or we can actually divide interphase into three distinct stages. We have G1, S, and then G2. The first stage then is G1. The G stands for gap 1. Personally, I tend to think of it just as growth. So G, although it stands for gap, I think of it as growth because during G1, the cell is literally just growing and it's doing its job. So whatever this specific cell is, if it's a skin cell, it's being skin. If it's a muscle cell, it'll be contracting. And then we go through G1 then. So the cell is just growing and doing its job. Then, once it reaches a certain size or age, it will enter into what is called the S stage of interphase. And this is really important because we'll look at this as we go through. But if you think about what mitosis is, you start off with one cell, and that one cell will divide and produce two new cells. But those two new cells, those two daughter cells, need to be identical to each other, and also they need to be identical to the parent cell. So that means the DNA specifically that we've looked at in previous sessions, so if you remember, DNA carries the instructions for the cell, particularly for producing proteins. Well, the DNA needs to be the same in both of those daughter cells. But for that to happen, you need to produce two copies of your DNA. So one copy can go into one daughter cell, and then the second copy can go into the second daughter cell. So before mitosis can begin, we need to replicate our DNA. So we can split it in half, then one half will go into one cell, one half will go into the other. And this is where that happens. So G1 is growth. We then enter into the S stage of interphase where we get the DNA synthesis. That is where our DNA will replicate itself. And we'll talk more about this in the coming slides. but essentially it gives us 2 copies of our DNA. We then move into G2, and G again just stands for gap, but again I think of it as growth, because in G2 the cell will complete its growth, continue doing its job, and then the only other thing that happens during G2 is that one of the cell organelles that we looked at in a previous session, called the centrosome, if you remember from that session on the cell structure, the centrosome is an organelle that's important in cell division. It organizes microtubules. For cell division, we need 2 centrosomes. During G1 and S, the cell will only have 1 centrosome. So during G2, the centrosomes replicate. That's about the only exciting thing that happens during G2. So we're just growing, we're doing our job, and the centrosomes replicate. And then once the cell is at a certain size or certain age, it will then be ready to divide itself. And it will then enter into mitosis. It will start to divide, and then it finishes off with this process called cytokinesis, which is the physical separating of the cytoplasm, or the actual breaking apart of the one cell into 2 cells. And then each daughter cell will then enter interphase, will enter G1 of interphase and start its very own cell cycle. Okay, so that is the life of a typical somatic cell. Before we begin looking at mitosis itself, I think it's worth spending a little bit of time actually looking at chromosomes specifically, because as we go through and talk about both mitosis and in the next session meiosis, what we're really doing is following the genetic material inside the cell. So although we know that there are a lot of other structures inside the cell, so things like the endoplasmic reticulum and the mitochondria, the Golgi body, all of those things, we don't really talk about those in mitosis and meiosis. They are just sort of getting divided and separated and shared out during the processes. What we're really focusing on is the genetic material, the DNA inside the nucleus, and how or what happens to that during these processes. So let's have a little closer look at chromosomes then. Chromosomes consist of DNA that is highly folded around proteins that are called histones. So we know that we know what DNA is now. It's a nucleic acid. It's formed of 2 strands of long chains of nucleotides forming a double helix. So if we look at the diagram on the right hand side here and we look right at the top, we recognize the structure of DNA. We see the double helix there. So we've got 2 long chains, 2 long polynucleotides joined together through those hydrogen bonds, and they sort of coil up to make this double helix. Well, inside each of our cells, we've got about 2 meters worth of DNA. So that is an incredible amount of material to fit into such a tight space. So how does that happen? Well, it happens through a really complex process of folding, twisting, and coiling, specifically using a special type of protein called histones. So if we go back to our diagram, so we've got DNA at the top. If we move down to the next one, we can see that these little sort of pink circles here, well, those are our histone proteins, and they act a little bit like a spool, and the DNA will wrap around those like cotton does around a spool. So when that happens, we end up with this sort of configuration of DNA highly sort of coiled around on these histone proteins. And when they're in this position, they are referred to as chromatin. Chromatin is the state that DNA is in when our cells are going through interphase. But as we start moving towards mitosis, our chromatin will start to condense, and it will start to form a much more dense structure that will make division a lot easier. You can see the different stages of that as we work down through this diagram. During interphase, chromosomes are unraveled and exist as chromatin. So we can see the chromatin in these 2 parts of the diagram here. But before the cell begins to divide, we go through, well, as we know, we go through DNA replication during the S stage of interphase. And then we go through G2, and then we start moving towards mitosis. Chromatin will begin to condense. And you can see it here, how the chromatin will start to sort of fold back over itself. And then it will fold back over itself again. And then it will start to twist and coil whilst also folding back over itself again. So you're getting this really complex arrangement of twists, coils, and folds until ultimately we end up with what we would recognize as the classic chromosome shape, this X shape. Don't forget what happened during the S stage of interphase. Our DNA replicated. So when we look at a chromosome like this, what we can see is this top part here and this bottom part here. These are what we would refer to as sister chromatids. So this is one complete copy of the DNA for that chromosome. And then under here, we've got the other copy that was produced during the S stage of interphase of the same chromosome. So these two are exact copies of themselves. So we refer to them as sister chromatids. And you can see that they are joined up here at a part called the centromere. This top part is one chromatid, and then this bottom part here is the other chromatid. So they are sister chromatids because they are identical copies of each other that were created during the S stage of interphase. And the two sister chromatids are connected at the centromere. So those are our chromosomes. So our DNA that exists in the nuclei of our cells. When the cell is going through interphase, it exists unraveled as chromatin, so DNA wrapped around these histone proteins. And as we enter into mitosis, that chromatin will go through this complex process of condensing, coiling, folding, and twisting until it is fully condensed into what we would recognize as a chromosome. And our chromosomes will consist of two sister chromatids. So this brings us on to the next bit, which is how many chromosomes do we actually have? So we'll look at that on the next slide. When it comes to the number of chromosomes that an individual has, it's important to know that different species will actually have different numbers of chromosomes. As humans, we actually have 46 chromosomes. So a typical human somatic cell, so if you remember, a somatic cell is any of our body cells other than our gametes, so other than eggs and sperm, and those cells will have 46 chromosomes, or we could say more accurately that we have 23 pairs of chromosomes. The reason that we say that we have 23 pairs is because as sexually reproducing organisms, we get a set of chromosomes from our mum and we get a set of chromosomes from our dad. So all of the instructions for building a human, so for building you, will come in a complete set of 23 chromosomes, but each of our somatic cells actually has two sets of chromosomes, one from our mum and one from our dad. So in total we have 46, so 23 pairs or two complete sets of 23, depending on how you want to describe it. And like I said, different species will have different numbers of chromosomes. So as some examples, mosquitoes typically have 6 chromosomes, a carrot will have 18, giraffes have 30 chromosomes, so 15 pairs. So as humans though, we have 46 chromosomes in our somatic cells, two complete sets of 23, or alternatively you could say 23 pairs of chromosomes. If we look at the picture here down on the bottom right hand side, we can actually see our 23 chromosomes, and they are numbered in order of size. So chromosome number 1 is the largest of our chromosomes, and then we move on to number 2, number 3, number 4, and so on, all the way down to the smallest of our chromosome pairs, which are chromosome number 22. And then we have a pair of sex chromosomes, so X and Y. So if you are female, you will have two X chromosomes for sex chromosomes, so you'll be XX. Or if you're a male, you will have one X and one Y chromosome. And if you look at the picture here, you can see that the Y chromosome is much, much smaller than the X chromosome. And each one of these chromosomes will be made up of millions of nucleotides. So if you remember what a chromosome is from the previous slide, it is a long strand of DNA wrapped around those histone proteins. And each of those chromosomes will be made up of millions of individual nucleotides all joined together through those phosphodiester links. And then you end up with the two strands of DNA that join together through hydrogen bonds to make the DNA double helix. So we have 46 of those different chromosomes, and we would say that we have two complete sets of 23. One set that we got from our mum and one set that we got from our dad. So again, if we look at this diagram here, this picture, we can see that we have two chromosome number 1s, two chromosome number 2s, two number 3s, and so on and so forth all the way through. So we take chromosome number 3 here, we would have got one of these from our mum in the egg and one from our dad in the sperm. And we actually give these names. So the two chromosomes that we get each pair are called homologous chromosomes or homologs. And homo means the same. So what we mean by this is if we take chromosome number 1, for instance, well, the one that we got from our mum and the one that we got from our dad, they will carry genes in the same locations on those chromosomes that code for the same proteins, essentially, or they code for the same characters in an individual. So this thing about characters and traits, these are terms that you might be familiar with, and these are something that we'll investigate in a coming session where we look at Mendelian genetics. For now, we can say that something like hair color would be a character. So the fact that you have hair and it has color would be a character. And then a trait would be the color that your hair is. So brown hair would be a trait. Blonde hair would be a trait. So that's the difference between character and trait. And we'll investigate that a little bit more in detail in a coming session. But for now, so you know, the homologous chromosomes are the matching pairs. So one we got from our mum, one we got from our dad, and they will carry genes. So a gene is just a section of a chromosome that carries all of the instructions for building a particular protein. So that will give us one of our characters. So the homologs will carry the genes for the same character at the same location. So we can, if we look at the diagram on the left-hand side here, what we can see is a homologous pair of chromosomes. So we could say that the left one we got from mum, the right one we got from dad. Typically, one chromosome will carry anything from a couple of hundred genes to a couple of thousand genes. So chromosome number 1, which is our largest chromosome, it carries about 2,000 genes on it. When we look at each one of these chromosomes, we can see that they have been replicated, so they have gone through the S stage of interphase, and now each chromosome consists of two sister chromatids. Okay, so you can see one chromatid here on the left, one chromatid here on the right, and then when we look at its homologue, we can see that that has also been replicated, so each of these chromosomes consists of two sister chromatids. So that is the homologous pairs of chromosomes. So you see it there in the text. Chromosomes in a homologous pair are the same length and carry genes controlling the same inherited characters. And we get one of our homologs from our mum and one from our dad. So hopefully that makes a bit of sense. And when we go through the processes or the process of mitosis, and later on when we look at meiosis, what we're going to be doing is we're going to follow these chromosomes and what happens to them when the cell divides. But essentially what we're going to do with mitosis is we're going to separate the sister chromatids of each chromosome, and then one chromatid will go into one daughter cell and then the other will go into the other daughter cell. So that's the main aim of mitosis. So we're going to look at that in a sec. So what we can see on this slide is an example of, we could just look at it, it's an organism that would have 6 chromosomes. So I mentioned just now that the mosquito, or many species of mosquitoes, but a lot of them will have 6 chromosomes. And we can write that down. So what we would say is that one complete set of chromosomes in a mosquito would be 3. So they're sexually reproducing organisms. So they have 2 sets of chromosomes, one from the male, one from the female. And if we take one of those complete sets, well, that would be 3. So they've got 2 sets of 3, giving them 6 in total. So if we wanted to write down the chromosome number for this species, what we would say is that N, so in biology and ecology, you often see the letter N that just denotes a population or something like that, or a number of something. So N equals 3. What that tells us is that a complete set of chromosomes for this species is 3. All of the instructions for building that organism are contained within 3 chromosomes. But because we get one set from the mother and one set from the father, so the maternal set of chromosomes, N equals 3, the paternal set of chromosomes from the male, N equals 3, and then we join those together in the somatic cells, and then we would write it like this. We would say that 2N equals 6. So one set is 3, another set is 3. Together, we would say that 2N is 6. So using that, just quickly jot down how you would denote that for humans. So what would N equal? And then what would 2N equal? So I'll just give you a second to write that down. Okay, so hopefully what you wrote down for that was that in humans, N would equal 23. So our maternal set of chromosomes, so the one that we got, the chromosomes we got from mum, N would equal 23. And paternal set of chromosomes, so the ones we got from our dad, N would equal 23. So that would mean in our somatic cells, we would have 2N equals 46. Okay, so hopefully that's what you got. N equals 23, 2N equals 46. And we can actually assign proper names to this. What we would say, if you had a cell, and our gametes are examples of this, cells that have just one set of chromosomes, and we'll look at this in the next section. Our gametes, our eggs and our sperm, have just one set of chromosomes, so they would be N equals 23, and we would say that they are haploid. Gametes are haploid. They have just one set of chromosomes. And then our somatic cells, which are 2N, are what we refer to as diploid. Somatic cells are diploid. They have 2 complete sets of chromosomes. Our gametes are haploid. They have just one set of chromosomes. So don't worry too much about that for now. These are terms that I'll introduce to you a bit more detail as we go through. So what we can see on this diagram here then, let's look at the actual words. So 2 sister chromatids of 1 replicated chromosome. So we know that this is showing, we've got in red the maternal set of chromosomes. So N equals 3. So we've got 1, 2, 3. And then we can see, as noted here, that these chromosomes have already been through the S stage of interphase. So they've already been replicated. So each chromosome consists of 2 sister chromatids, which we can see there as the 2 different lines there. And remember, our sister chromatids are joined at the centromere. And then what we can also see here is because we've got 3 maternal chromosomes and 3 paternal chromosomes, That means that we have 3 pairs of homologous chromosomes. So we can see that here. These 2 are the same length. They will carry genes for the same characters at the same locations. So we can say that these are a homologous pair of chromosomes. This blue one and this red one, well, this would be a homologous pair of chromosomes. And then these 2 here, this red and this blue, this is our 3rd pair of homologous chromosomes. And this last one down here, this label here, so 2 non-sister chromatids in a homologous pair. Now that sounds like a real mouthful, but we can break it down and look at what it actually means. So what we've got here is a homologous pair of chromosomes. So the red chromosome here was from mum, the blue one here was from dad. We know that these have already been through the S stage of interphase. So each chromosome consists of 2 sister chromatids. What we could say though is if we take this sister chromatid here of the paternal chromosome and we refer to it or compare it to this sister chromatid from the maternal chromosome, we would actually say that these are non-sister chromatids of the homologs, or non-sister chromatids of the homologous pair. So hopefully that makes sense. They still carry the genes for the same characters in the same locations, but it just so happens that this one is from the maternal chromosome and this one is from the paternal chromosome. That will become particularly important when we start looking at meiosis in the next session. But for now, we can just remember that one set of chromosomes would be classed as N, and we would refer to that as being haploid. So for humans, N equals 23, and that would be haploid. 2N is 2 complete sets of chromosomes, so 2N in humans would be 46, and that would be diploid. That gives us the terminology and the tools to start looking at mitosis, so we can start looking at cell division. Mitosis itself consists of 6 stages, or mitosis proper actually consists of 5 stages, and then we have the final 6th stage at the end, which is slightly different. But let's go through them quickly, and then I'll go through each one and tell you a little bit about what happens during each stage. And then I've got an animation to show you at the end, which will hopefully illustrate it and really help you to understand it. So we start off, so we've moved through interphase now. We've gone through G1, we've gone through the S stage of interphase where our DNA is replicated to give us our 2 sister chromatids. We've gone through G2, and now we enter into mitosis. And we start off with something called prophase. We then move into prometaphase. So we go from prophase into prometaphase, into metaphase, and then we move on to anaphase, and then we move into telophase or telophase. So those are our 5 stages of mitosis proper, and then we have a final stage here, which is cytokinesis. Now in some texts that's classed as a stage of mitosis. In other places it's a sort of standalone stage, but I tend to keep them all together. It keeps it nice and simple. So those are our stages of mitosis, during which our cell is going to divide into 2. So we'll start off with a parent cell, and then at the end of mitosis, we will end up with 2 daughter cells that are genetically identical to each other and the parent cell. So let's go through each of these stages, and we'll talk a little bit about what happens during each one. Mitosis begins with prophase then. And on the diagram here, we can see what's starting to happen. So inside the nucleus here, we can start to see that our chromatin is condensing. So we're starting to see the formation of our chromosomes. So we're not following a full set of chromosomes here. We're only following 4 by the looks of it in this one. It'd be a bit messy if we were following the full amount of 46 for us. But what we can see is that our chromatin is starting to condense. And then what we can also see up here is This is a centrosome. And then down here we can see our second centrosome. So you remember that they replicated during G2 of interphase. And centrosomes are microtubule organizing centers. So if you remember, microtubules are part of the cytoskeleton. So they are these hollow tubes that are used for sort of heavy shifting work inside the cell. And microtubules begin to grow out of the centrosomes, and then they will actually meet each other in the middle of the cell, and they start to form something called the mitotic spindle. Okay, so this network of microtubules growing out of the centrosomes is called the mitotic spindle, and that is starting to grow during prophase. But at this stage, we can still see that the nuclear envelope is still in place. So we've still got the membrane around the nucleus. We also still have the nucleolus, which you can just sort of make out up here. Remember, that's the area of the nucleus where ribosomes are manufactured. So all of that is still in place. But this is prophase then. We're starting to the cell is starting to get ready to divide. So the chromosomes are starting to condense, and we're starting to get the formation of the mitotic spindle. That will then take us into prometaphase. So what we can see now is that our discrete chromosomes finally appear. So what we can see here now is what we would recognize as proper chromosomes. So again, as I said earlier on, or just now, we're only following 4 chromosomes here. So n equals 2, 2n equals 4. And each chromosome consists of 2 sister chromatids. And we say that we've got 2 homologous pairs. So this would be a homologous pair. This would be our second homologous pair. And we recognize these as fully formed chromosomes now. The process of condensing has happened. We can also see that the nuclear envelope has started to disappear. So it's starting to fragment. So there'll be enzymes at work there breaking down the nuclear envelope. And then what we can also see is that our mitotic spindle has grown considerably. So the microtubules growing out of the centrosomes have now pushed the centrosomes to opposite sides of the cell, what we would refer to as opposite poles of the cell. And the next thing that will happen is some of these microtubules coming out of the centrosomes will begin to attach to the chromosomes. So I mentioned to you previously that our sister chromatids of each chromosome, so if we take this chromosome up here, we can see that each chromosome consists of 2 sister chromatids. Those chromatids are joined at the centromere. Now, located at the centromere, we've got these little patches of protein called kinetochores. Don't worry about this too much. Don't let it ruin your life. But it's just more words for you to be aware of. These kinetochores are just little patches of protein located at the centromeres of each chromosome, and microtubules will attach to the kinetochores. Okay, so what that will mean is the microtubules can now start moving the chromosomes around because they are attached to the chromosomes. Where do they attach? They attach to kinetochores located at the centromeres. So many words. But that's where we are with prometaphase then. Our mitotic spindle is pretty much complete. The nuclear envelope has broken down. Our chromosomes are fully condensed now. Microtubules are attached to the chromosomes, and then that means that they can now start moving the chromosomes around. And that is exactly what we see in the next stage, which is metaphase. When we get into metaphase, our mitotic spindle is now fully complete. And what we can see is that as the mitotic spindle is completed, it's shifted the chromosomes so that they are all aligned along the middle of the cell. So we refer to this sort of imaginary line or this imaginary equator through the middle of the cell as the metaphase plate. So you can see that up here. It's not a real plate, obviously. It's just an imaginary line through the middle of the cell, and the chromosomes are aligned along it. And they are aligned in such a way that each chromosome has one of its sister chromatids facing towards one pole of the cell, and the other sister chromatid is facing towards the other pole of the cell. So we can see all four of these chromosomes are arranged in that way. Now, clearly, if this was one of our somatic cells, instead of having 4 lining across there, we would have 46 lining across there. That would be a little bit messy to put in this diagram. But metaphase is where we have our chromosomes aligned along the middle of the cell. So the chromosomes attached to microtubules at their kinetochores are all aligned along the metaphase plate. So what we are now in a position to do is separate or split apart the sister chromatids. So you can imagine it in a second. What's going to happen is these two sister chromatids will be pulled apart. One sister chromatid will move up here, and then the other sister chromatid will move down here. And that's exactly what we see in the next stage, which is anaphase. So in anaphase, the chromatids of each chromosome have separated. So essentially what is happening is that the microtubules begin to shorten. And as they shorten, they pull the sister chromatids apart from their centromeres. So we can see here we've got one set here of chromatids going up to this side of the cell and the other moving down to this side of the cell. Now, this again is where our terminology gets a little bit strange. Let me go back to this one. At this stage, we would say that we have 4 chromosomes, but each chromosome consists of 2 sister chromatids. Okay, so we've only got 4 chromosomes. When we move into anaphase, once the sister chromatids have been separated, once they've been pulled apart, they each become their own distinct chromosome. So what we would say now in this picture, we can actually see 8 chromosomes. So we've got 4 up here and 4 down here. So again, it's just the way that the terminology works. So whilst the sister chromatids are attached to each other, they will be classed as one single chromosome, but that chromosome consists of 2 sister chromatids. As soon as you pull those sister chromatids apart, each chromatid will now be classed as its own individual chromosome. Hopefully that makes sense. So within anaphase, then, our microtubules start to shorten, pulling the sister chromatids apart. And these daughter chromosomes, as we'll call them, start moving towards the opposite poles of the cell. So you can also almost imagine the centrosomes acting a bit like a reel of a fishing rod. They start reeling in these daughter chromosomes, pulling them down towards the poles or pulling them up towards the pole of the cell. Once that's completed, we can then move in to telophase and then cytokinesis ultimately. So within telophase, so we can see that here. So the reason I've put these together is they typically will start to happen around the same time. So once anaphase is completed, the daughter chromosomes are moved to the poles of the cell. Those chromosomes will start to decondense. They will start to unravel back to chromatin, and we can start to see here the reformation of the nuclear envelope. And then at the same time, cytokinesis will typically begin. And cytokinesis is a process by which contractile proteins start to form around the middle of the cell, and it kind of pinches the cytoplasm apart, basically pinching the parent cell into these 2 daughter cells. I always think of this as a bit like a drawstring on a laundry bag or something like that, where you can pinch the top of the bag together. This is kind of what's happening with these contractile proteins. So we start off with this something called the cleavage furrow, and then that will continue to grow inwards until ultimately these 2 will pinch apart, and that is essentially what cytokinesis is. So cyto refers to cell, and kinesis, sort of, you can use that to break apart. So it is splitting apart the cytoplasm. And then at the end of it, we end up with two separate and distinct daughter cells that are genetically identical to each other. Both of those cells will be diploid. So what we see with mitosis is that it conserves the number of chromosomes. So because we duplicated or replicated our chromosomes during interphase, so each chromosome consisted of two sister chromatids, what we've done during mitosis is we've just separated those sister chromatids. So we end up at the end of mitosis with two daughter cells that are diploid and they are genetically identical to each other. So I wanted to show you this animation. So this is from a company called NDSU Virtual Cell. So this is available independently or separately on YouTube. And it's a really, really good animation that shows the process of mitosis. And they also do one for meiosis that we'll look at in the next section. What the narrator will do, she will go through this quite quickly, and then she will slow down and go through it in more detail, and then she will summarize quickly again at the end. So don't panic when she moves through it quickly at the beginning. So let's go through, take a look at this, and we'll catch up at the end.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
So hopefully that was useful. It's worth going over that a couple of times, or a few times even. As the more times you watch that animation and you go through this session, it will really help to solidify the key points in your memory. So on that note, let's do a quick summary of mitosis then, looking at the key points of it. So mitosis is the division of somatic cells. So remember, those are our body cells, any cell other than our gametes or eggs and sperm. It is important for development from a fertilized egg. Once we're born, it's important then for growth to maturity. And then throughout our lives, it's important for repair. Well, what does mitosis produce? Well, it produces two daughter cells that are genetically identical to each other. And they'll also be identical to what was the parent cell that obviously doesn't exist anymore because the parent cell has divided into two daughter cells. You can see on the diagram at the bottom here. And the last thing to mention is that mitosis actually conserves the chromosome number, say the number of chromosomes. And that is because during the S stage of interphase, we replicated or duplicated our genetic material. So the chromosomes were replicated. So now at the end of the S stage of interphase, each chromosome consists of two sister chromatids. So we start off mitosis with 46 chromosomes, and then we finish mitosis with two daughter cells that also have 46 chromosomes. So we say that the parent cell is diploid, so it has two complete sets of chromosomes, and then each daughter cell is also diploid. So hopefully that makes sense. So as always, we'll do a quick recap quiz to see what you remember. So I'll read through the questions and I'll give you a few minutes to answer them, and then we'll go through the answers together. So question number 1, how many daughter cells does mitosis produce? Question 2, what is mitosis used for? Number 3, how many chromosomes do humans have? Number 4, during which stage are the sister chromatids separated? And then number 5, when do the centrosomes replicate? So 5 questions. I'll give you a few minutes to answer those, and then we'll go through the answers together. Okay, so hopefully you've had a chance to answer those. So let's go through the answers. So question number 1, how many daughter cells does mitosis produce? Well, it's 2. Number 2, what is mitosis used for? It's used for development, growth, and repair. Number 3, how many chromosomes do humans have? Well, you could have worded this in different ways, but we have 46 in total, and that is made up of 2 sets of 23 or 23 pairs, depending on how you want to word it. But the important bit is we have 46 in total, made up of 2 sets, 1 maternal and 1 paternal. Number 4, during which stage are the sister chromatids separated? Well, that's anaphase. Then number 5, when do the centrosomes replicate? G2 of interphase. So hopefully you did okay with those. If not, don't worry too much at this stage. Just go back over it a few times. I do appreciate that it's one of those topics where if this is the first time you've done it, I've just introduced you to a lot of new terms. So hopefully you took note of what I said at the beginning and you've written down those words, and then hopefully you've been able to write down some definitions as we've gone through. And it's just worth going through them a few times and testing yourself. So write down on a blank sheet of paper or on a blank Word document or something like that, write down homologous chromosomes and then write a definition of what homologous chromosomes are. It might take a few goes for you to get it, but you practice that a few times and it'll really help it to sink in. Unfortunately, there's no shortcut to learning this stuff. It's just a case of sitting down and sort of plugging away at it, going over it a number of times, testing yourselves and just practice. So that brings us to the end of this session on mitosis then. So as we've gone through this, hopefully by the end now, you should be able to explain why mitosis is important in growth and repair. You should also be able to describe the main stages of mitosis in somatic cells, and then also explain how mitosis conserves the number of chromosomes from parent to daughter cells. Okay, so hopefully that was useful. And I'll see you in the next session where we will look at meiosis, so the production of gametes. Okay, thanks very much.